In this video, we will study about the DNA replication. DNA replication is the biological process of producing two identical replicas of the DNA from one original DNA molecule. And as simple as it may seem, it is one of the most complicated biological processes and involves the accurate copying of almost 6.5 billion base pairs in a complete set of DNA during each cell division. DNA replication takes place inside the nucleus of the cell, where two identical and new copies of the DNA are synthesized from the original DNA. The newly formed set of DNA will then be transferred to the future nucleus that is produced during the process of cell division. At the end of cell division, we have two completely new cells, each having its own set of DNA. Now, in the previous videos in this section, we discussed in detail about the structure and function of the DNA, about nucleic acids, and we also talked about the concepts of complementary base pairing and directionality. These concepts are essential for understanding the process of DNA replication, so make sure to watch both these videos for a better understanding of this section. Now let's talk about the overview of the process of DNA replication. What you see here is the simple double helix model of the DNA. In this model, you can note that it consists of these two blue lines which basically represent the sugar phosphate backbones of the DNA, and at the center we have these orange lines which represent the nitrogenous base pairs of the DNA. If we untwist this double helix model, we will get something which is known as the step-ladder model of the DNA, which is a simple depiction of the structure of the DNA. In this example, you can see that the DNA consists of these two identical strands which almost run parallel to each other. Now, in the process of DNA replication, what happens is that these two strands separate. The original DNA which separates to form these two strands is called the parent DNA. And then these two strands which are separated from each other act as templates upon which new strands are copied and formed. Now, this process is made possible by the concept of complementary base pairing. Basically, we discussed the process of complementary base pairing in detail in the previous videos, and in a nutshell, you can note that the DNA consists of 4 nucleobases, which are the adenine, cytosine, guanine, and thymine. In the structure of the DNA, the base adenine always pairs with the base thymine, and the base guanine always pairs with the base cytosine. This is called the Chargaff's rule of DNA complementary base pairing. This is done so to maintain a proper distance between the two strands and also maintains a proper stable structure of the DNA helix. When the parent DNA is split, each strand acts as a template for the formation of new strand. This leads to formation of two completely new sets of DNA which are called as the daughter DNA. Now, if you take a closer look at this step-ladder model of the DNA, you can note that it consists of these two strands which are joined together in the center by hydrogen bonds. Now, these two strands run in completely opposite direction to each other, which means they are anti-parallel, and they also have a direction. Now, in DNA, we don't have the north, south, east or west directions, but on DNA, we have two ends, a 5' end and a 3' end of this strand. And since the strand above is running in completely opposite direction, it will have its 3' end on this side and its 5' end on this side. So due to this strand below is said to be running from the 5' to 3' direction, and the strand above is said to be running from the 3' to 5' direction. Now, to understand the concept of direction in DNA in a better way, let's take a brief and close look at the structure of the DNA. Now, in this example, you can see the different nucleotides and how they are joined together in one strand of DNA. If we take one nucleotide out from this strand, you can see the simple depiction of the chemical structure of a nucleotide. We will not go into the details as we have discussed the structure in detail in previous videos, but you can note that we have 3 important groups in a nucleotide, which are the phosphate, the sugar, and the nitrogenous base. Now, if we isolate the deoxyribose sugar from this nucleotide, we can see that the carbon atoms in the deoxyribose sugar are numbered from 1 to 5 in a clockwise manner, with the 5th carbon outside the ring. Now, you can compare this illustration with the chemical structure of deoxyribose for a better clarification. And it is the 5th carbon to which the phosphate group is always attached. Now, if we take a look at the structural illustration of a single strand of DNA again, you can note how the deoxyribose sugars are arranged in this single strand. And you can also see how all the deoxyribose sugars are aligned in this direction. Due to this orientation, the 5th carbon is always located on this side to which the phosphate groups are attached, and the 3rd carbon are always located on this side to which the hydroxyl group is attached. This defines the 5' end and the 3' end of this strand of the DNA. Now if you take a look at the second strand of the DNA, you can note the key difference, that is that it runs in completely opposite direction, which you can clearly tell by looking at the orientation of the sugar molecules in this strand. These sugar molecules are aligned in this direction, which is completely opposite to the strand below. And this defines the 3' end and the 5' end of this strand. Now the question arises, why is the concept of direction important in DNA replication? Well, this is because the main enzyme of the DNA replication, which is DNA polymerase, it can only add new nucleotides as a 3' end of the DNA. This means that the process of DNA replication can only progress from the 5' end to the 3' end of the DNA. This is because the new nucleotides can only be added at the 3' end. Well, we will get into the details of the concept of direction in DNA replication later in this video. Now let's talk about the various enzymes that make this complex process of DNA replication possible. In this table, we will study about the name, the symbol, and the function of the enzyme involved in the process of DNA replication. The first enzyme that we will discuss is the enzyme helicase, which will be denoted by this symbol. Now, the main job of the enzyme helicase is that it unzips the double helix structure of the DNA. The enzyme helicase performs this function by breaking the hydrogen bonds between the base pairs. This separates these two strands, creating the replication fork upon which various other enzymes will act in the process of DNA replication. So the next enzyme we will discuss is the enzyme DNA polymerase, which will be denoted by this symbol. The DNA polymerase performs the vital function of formation of a new strand of DNA known as the daughter strand. Now in this example, you can see the replication fork created by the enzyme helicase, and here is the 5' end on the left and the 3' end on the right of the upper part of the DNA. The DNA polymerase performs the function of formation of a new strand by arranging new nucleotides in the form of a DNA strand by using the parent DNA as a template. Now you can also note that the DNA polymerase cannot synthesize a brand new strand but requires a small sequence of RNA to do so. This action is performed by another enzyme. So the next enzyme which we will discuss is the enzyme RNA primase. The RNA primase will be denoted by this symbol. Now, like we just discussed, the DNA polymerase cannot initiate the synthesis of a new strand of DNA from scratch. So the enzyme primase synthesizes a short segment of RNA known as the primer, which acts as a guide for the DNA polymerase to synthesize the strand further. Now this short segment of primer is later removed and replaced by a new segment of DNA. The next enzyme we will talk about is the DNA ligase, which will be represented by this symbol in the process. The DNA ligase performs a vital function of joining together of the Okazaki fragments on the lacking strand. Now this may seem complex right now, but we will get to this in detail later in this video. And the final enzyme we will talk about is the enzyme topoisomerase, which will be denoted by this symbol. Now, the DNA is a double helical structure which consists of two strands of DNA which are twisted around each other. When the enzyme helicase opens up the DNA, there is generation of stress in DNA strands which can lead to supercoiling of DNA. Now, this process is solved by topoisomerase, which cut and reseal the fragments of DNA to relieve the stress that is produced by unzipping of the DNA. So this was part 1 of this video, in which we discussed the overview, the concepts of directionality, and the various replication enzymes involved in the process of DNA replication.